Good morning, class. Today, we're going to uncover one of the greatest secrets of life, DNA replication.
Oh, you mean how our DNA copies itself?
Exactly. Without DNA replication, cells wouldn't divide and life wouldn't continue.
That's because every new cell needs a copy of DNA, right?
Correct. But the real question is, how does DNA make an exact copy of itself?
Ooh, is it like a photocopier?
Not quite. A photocopier makes a flat copy. But DNA is a twisting, spiraling double helix, and it has to be copied perfectly, without errors. It's more like a factory assembling new DNA. Let's imagine we are tiny workers inside a cell, watching DNA replication happen. Picture DNA as a giant twisting ladder. The first step in replication is to unzip this ladder.
How do we do that?
A special enzyme called helicase is like a zipper pull. It unwinds the DNA, breaking the weak hydrogen bonds between the base pairs.
So it's like opening a jacket?
Exactly. Once helicase unzips the strands, each half of the DNA is exposed and ready to be copied.
So now we have two half ladders floating around.
Yes, and each half will serve as a template for the new strand. Now that the DNA strands are separated, we need to build the missing halves. That's where another enzyme, DNA polymerase, comes in.
Oh, that's the enzyme that adds new bases, right?
Yes, DNA polymerase moves along each strand, adding the correct matching bases to build a complete strand.
But how does it know which bases to add?
Great question. DNA follows a strict rule. Adenine, A, always pairs with thymine, T. Cytosine, C, always pairs with guanine, G. So if one strand has ATCG, the new strand will have TAGC.
It's like a puzzle where only the right pieces fit together.
Perfect analogy, but there's a small problem. DNA polymerase can only build in one direction, from 5' to 3'. That means one strand is built smoothly, the leading strand, but the other, the lagging strand, has to be built in small fragments.
So it's like painting a wall. One side gets painted in one smooth stroke, but the other side needs little patches.
Exactly. The lagging strand is made in Okazaki fragments, which are later glued together by another enzyme called ligase.
Wait, what if polymerase makes a mistake?
Smart question. DNA polymerase has a proofreading ability. If it adds the wrong base, it removes and replaces it immediately. Now for the final step. Once all the bases are added, another enzyme, DNA ligase, acts like glue to seal the fragments together.
And now we have two identical DNA strands?
Yes, one old strand and one new strand in each. This is called semi-conservative replication.
Semi-conservative means each new DNA keeps half of the original?
Exactly. That way, DNA is preserved accurately across generations.
So, let's recap. Helicase unzips the DNA. DNA polymerase builds new strands, matching the bases. The leading strand is built continuously, while the lagging strand is built in fragments. Ligase seals everything together.
Perfect summary. Now, imagine this process is happening millions of times every second in your body.
Whoa, so my body is basically a giant DNA copying machine?
That's right, and that's how life continues, one perfect replication at a time.
I'll never look at DNA the same way again. Me neither. This was awesome.
Thank you. Meet you all in the next class. Goodbye, students.